Hello, my name is Cleo Valentine, and I am a PhD candidate at the University of Cambridge with joint supervision between the Department of Architecture and the Computational Neuroscience Group. In this lecture, I'm going to be presenting the findings from a pilot study that I conducted, which examined the impact of biophilic architectural design on neuroinflammatory activity using quantitative EEG. This research really sits at the intersection of architecture and neuroimmunology. So more specifically, my research examines how architectural form impacts physiological stress and how that in turn may mediate neuroinflammation or inflammation of the brain. Neuroinflammation is heavily implicated in the development of several neurodegenerative and psychiatric disorders. For example, as of 2021, neurodegenerative conditions were the fifth leading cause of death globally. Globally, the number of people living with dementia is predicted to increase from 50 million in 2018 to 152 million in 2050, which is a 204% increase. Moreover, the cost of dementia is expected to almost double by 2050, from £25 billion in 2021 to £47 billion in 2050. So I'm really interested in how the built environment may contribute to the development of these conditions, but also how design could potentially be used to help combat them. Before I get too far into this presentation, I would like to clarify a few terms. First, I refer to the term physiological stress or stress a fair bit, and by this I mean any threat, real or implied, to one's physiological or psychological integrity. Stress events can range from physically traumatic accidents, such as a car accident, to a confusing or a disorienting spatial layout, such as many hospitals. I also use the term architectural forms. In this research, I use the term to refer to subtle variations in the shape or configuration of the built environment, which has been my primary research focus to date. Emerging evidence from the field of neuroarchitecture, which examines the impact of architectural form on the brain and nervous system, has found that exposure to certain architectural forms can impact stress levels, and as a result influence health and wellness. For example, the health benefits of biophilic or biomorphic design are well documented. Biophilic design refers to the direct incorporation of organic material into design, for example, incorporating trees or greenery into a facade, whereas biomorphic design refers to the integration of naturalistic geometries into design, for example, the leaf-like structures on Corinthian columns, or the incorporation of certain aspect ratios. For the purposes of this presentation, I'll largely use the terms interchangeably. Biophilic design has been associated with reductions in physiological stress responses, specifically increased frontal alpha wave activity, reduced blood pressure, and skin conductance. Whereas exposure to urban views, and by that I mean views of skyscrapers, for example, has been associated with increases in physiological stress responses, specifically decreases in frontal alpha wave activity, reduced blood pressure, and skin conductance. While there's compelling evidence supporting the positive effects of biophilic architecture on stress, to date, less consideration has been given to understanding the physiological outcomes associated with these responses. So by this I mean, what are the downstream effects of architecturally mediated stress reductions? However, the emerging field of neuroimmunology may be able to shed some light. Neuroimmunology is a relatively new discipline that examines the interplay between the nervous system and the immune system, both of which play a critical role in mediating the body's interaction with its environment, including the built environment. Neuroimmunological research suggests that physiological stress responses are not discrete singular events, but instead stress responses can set off an inflammatory cascade across various pathways within the body. This is a simple diagram from a recent paper of mine which came out in April of 2023 that maps the causal effects of a stress event. You can see that in an acute setting, a threat engages the body's fight or flight mechanisms, and this results in a number of changes across the body. These responses are designed to return the body to a state of equilibrium by providing the necessary physiological responses to allow the individual to evade or navigate the stressful situation. And this process is highly effective at regulating acute stress events. However, if the stressor becomes chronic, this can result in prolonged neuroendocrine responses, which actually compromise the immune system and can cause long-term damage to the organs and tissues, including the brain. Neuroinflammation refers to inflammation of the brain, and chronic neuroinflammation is associated with the development of a number of devastating neurological conditions, including psychiatric conditions such as depression, anxiety, and schizophrenia, and neurodegenerative disorders such as Alzheimer's disease, Parkinson's disease, and ALS. I focused on neuroinflammation as opposed to other forms of inflammation, because not only are the consequences devastating, but also the presence of these conditions are increasing exponentially in their prevalence, particularly in urban areas. To review, the connection between architectural form and physiological stress is well documented. Similarly, the connection between physiological stress and neuroinflammation is well documented. However, neither neuroimmunology nor architectural neuroscience has directly considered the potential impacts of architectural design on neuroinflammation. Just to clarify, it is not the case that all stress responses result in neuroinflammatory activity. However, given the strong connection between stress and neuroinflammation and the connection between architectural form and stress, it certainly seems worthy of further investigation. So in response, I designed and conducted a pilot study to examine the relationship between biophilic architectural design features and neuroinflammation. This study hypothesized a negative correlation between the level of biophilia and the level of neuroinflammatory activity. In other words, the study hypothesized that an increase in biophilic design features would result in a decrease in neuroinflammatory activity. Before I get into the details of my study, I always find it helpful to clarify the scope of this research. First, the results that I'm presenting here are the product of a pilot study. So this was an initial attempt to assess the feasibility of evaluating neuroinflammatory responses to architectural design using EEG techniques. Second, this research assesses subconscious and objective physiological responses to architectural stimuli. It does not evaluate conscious preferences or subjective evaluations of architectural forms. I'm interested in how your body responds, less interested in how you think you respond, because interestingly, these two aren't always aligned. Third, EEG is quite a new method for analysing neuroinflammation. As a result, a base level was assumed based on current literature, and the neuroinflammatory responses were measured using the difference between exposures. Moreover, I think it's really important to acknowledge how complex physiological systems are. There are a number of socioeconomic, environmental, psychosocial, and epigenetic factors that influence both the brain and the body. It's also important not to be reductionist. However, if architecture does play a role, in my opinion, anything that helps to support human health is a win. So, as part of this study, 10 participants' neuroinflammatory responses were assessed while they were exposed to images of architectural facades with varying degrees of biophilia. 2 separate buildings were evaluated. 3 images per building were displayed for 180 seconds for a total of 18 minutes. The level of biophilia in each respective building was measured using a pre-existing framework called the Biophilic Healing Index, developed by Nico Salangeros at UT Austin. What the BHI allows you to do is to evaluate the degree of biophilic features in a building and assign an empirical value to represent this level. Going forward, I'm looking to develop an objective computational approach for measuring architectural features, potentially building off a piece of research I did, which used machine learning image analysis to identify biomorphic geometries in architectural facades. The level of neuroinflammation was measured by examining specific types of changes in electrical activity within the brain using a 32-channel EEG device. These changes in electrical activity can act as a proxy variable for neuroinflammation. I was particularly interested in changes across the relative power delta band and the relative alpha band, which can be indicative of neuroinflammatory responses. Changes in these frequency bands can indicate changes in microglial cell activity, which are the brain's resident immune cells, which can trigger inflammatory cascades of cytokines such as interleukin-1, interleukin-6, and tumor necrosis factor alpha. 2 buildings were evaluated. The first building, the De Kroon building, served as the control, as according to the BHI, it does not incorporate a large degree of biophilic design features. The De Kroon is a mixed-use building, constructed in The Hague and designed by Rapp & Rapp Architects in 2011. The images were selected so that there was a high degree of similitude between the control and the dependent. Conversely, the Hyperion City building, developed by Wave 1 real estate developers in Berlin and designed by Studio Pret in Austria, acted as a dependent, as it intentionally incorporated biophilic design principles. The Hyperion City is a proposed mixed-use building that incorporates urban farming, greenery, and biomorphic geometries in an attempt to support resident health. I'm very grateful to Wave 1 for funding this research. This study produced 3 primary findings. First was a decrease in relative delta. This connection between biophilia and relative delta power is perhaps the most significant finding from the study, given the association with delta power and neuroinflammation. This finding supports the study's hypothesis. Second was that there was no change noted across the relative alpha wave activity. This result is surprising, given the association between biophilic design and relative alpha power in neuroinflammation. This may be the result of the protocols used, but it certainly needs to be investigated further going forwards. Lastly, was that changes were noted in the relative gamma band. This was unexpected, but is consistent with recent findings that suggest that relative gamma power may be a reliable proxy metric for physiological stress responses and may be associated with neuroinflammatory activity. It's important to note that this study faced a number of theoretical and methodological limitations, including but not limited to the 2D and unisensory nature of the stimuli, the sampling methodology, the data collection technique, specifically the EEG data processing method, and the research design, specifically statistical techniques that were used. However, I do hope that this methodology will become more refined as the research progresses. As I mentioned, this study is a pilot study, so it was an initial attempt to measure the impact of architectural design on neuroinflammatory activity. Future research may wish to go in a number of different directions. First would be to gain a better understanding of what the observed changes in EEG data indicate and how spatial forms create these neuroimmunological deviations. This would mean looking more closely at specific EEG frequency bands, but also drawing in findings from environmental psychology and evolutionary biology to paint a fuller picture. Second, it will be critical to understand what architectural forms elicit neuroimmunological responses. This will require controlled studies which can isolate individual features and test their respective impacts on neuroinflammatory responses. Third, it will be important to understand how much exposure is required to elicit these changes. For example, does someone need to spend a large portion of time in a space, or can they benefit or be harmed by momentary exposure, such as walking to work? Fourth, given the complexity of neuroinflammation, it will be important to disentangle how, if at all, individual stressors interact with one another to create either cumulative or net effects. For example, is it possible that stress-inducing environmental qualities, such as a crowded street, could be mitigated by the introduction of stress-reducing architectural forms? Currently, I am investigating the corollary of this study, namely whether certain architectural forms, particularly seemingly benign ones that we incorporate into design every day, may increase neuroinflammatory activity. Going forward, I'm hoping that architectural neuroimmunology grows as a discipline, and that the findings from these studies can help to inform the design and the construction of a healthier built environment. Thank you very much, and please don't hesitate to drop me an email or contact me via my website should you have any questions. Thank you very much.